Okay, so we're going to cover the Krebs cycle today. This is going to include the key reaction and the key enzymes, so literally everything you need to know for the Krebs cycle. So you have 8 reactions in total, and it can also be called the Citric Acid Cycle. So that means that if maybe it will be an exam or something, you might see that name, just know it's the same thing. So where is it located? It's located in the mitochondrial matrix. This is almost always asked, so make sure you know it. And why do blood cells not have the Krebs cycle? Now this is because they lack mitochondria, so they can't carry out the Krebs cycle processes. This is also quite a good question that could be asked. So let's move on to the first of the 8 reactions. So we're going to be converting acetyl-CoA, this is coenzyme A, and oxaloacetate into citrate. And quite an easy enzyme to remember is just citrate synthase because we're making citrate. So the second one is going to be citrate converting to isocitrate. And now this is the first reversible reaction. And this is going to be catalyzed by dehydration, then hydration. So it is 2 reactions really split into one, or simplified into one even. Just a simple isomerization. And the third one is isocitrate, which is converted to ketoglutarate. And now this one's going to be catalyzed by isocitrate dehydrogenase. And I would say that the third reaction of the Krebs cycle is very, very important. I'm going to put down the reasons why now. So first of all, we can say that NAD plus can be converted to NADH plus a proton. And this is happening in the cell when we want catabolic processes. And alternatively, NADP can be converted to NADPH plus a proton. And this is if the demands of the cell are anabolic. So like I mentioned, these differ depending on the demands of the cell. And this is because the process can be amphibolic, so it can be anabolic or catabolic. And again, this is a decarboxylation reaction, number 3. Let's move on to number 4 now, reaction 4. So we're going to have ketoglutarate, and that's going to be converted to succinyl coenzyme A. And this is catalyzed by ketoglutarate dehydrogenase complex. That is the enzyme for this reaction. And this reaction will give out CO2, carbon dioxide, which therefore does mean it is a decarboxylation reaction again, and also an irreversible one as well, as you can see by the way I've drawn the arrows. So this reaction consumes coenzyme A, it produces carbon dioxide and NADH and a proton. Let's look at number 5 now. Succinyl-CoA is going to be reversibly reacted into succinate. And this reaction is going to give out carbon dioxide, GTP, and coenzyme A. This reaction is reversible, and we're losing coenzyme A, and it's the only reaction that gives out GTP, which is an energetic molecule. So number 6, succinate converts to fumarate, and this is going to transfer 2 hydrogens to FAD. And this is a catabolic process because we are breaking something down to produce energy. Let's look at reaction 7 now. Fumarate is converted to malate. And we hydrate this reaction, so we're adding water in. And this is where the double bond that was previously formed in reaction 6 is now lost from the hydration with water. And finally, the 8th reaction, converting malate into oxaloacetate. Remember, oxaloacetate was the first substrate. So the final product is the first substrate because it's a cyclical reaction, the Krebs cycle. So we're going to convert NAD plus into NADH and a proton. This is a dehydrogenation reaction again, as the 2 hydrogens are lost. So hopefully that's made the Krebs cycle really, really easy. I hope you enjoyed.